Good morning students. In this class we will study about pathogenesis. So what is meant by pathogenesis? The whole array of plant reactions for the action of particular pathogen is called as pathogenesis. So there are different names for pathogenesis, that is disease cycle or infection cycle. So the definition of pathogenesis is the action of pathogen and whole array of host reactions for that particular pathogen and disease development occurring in host is called as pathogenesis. The disease development may be a continuous or intermittent process. So it is not a single process, it is a collection of numerous process. So the series of more or less distinct events occurring in sequence and leading to disease development and perpetuation of disease and pathogen is called as pathogenesis. So the perpetuation means continuation of disease or multiplication of disease or pathogen in particular plant or host. So types of disease cycle or types of pathogens, we can classify the pathogens or disease cycle as per the time period taken by particular pathogen. So pathogen may complete one or more disease cycles per year, or in contrary take more than 1 year to complete a single disease. So based on the time period taken by particular pathogen, we can classify monocyclic or single cyclic pathogens. So it will complete single cycle per season. Polycyclic or multicyclic pathogen, so pathogens complete more than 2 cycles per year. Then polytic pathogens. So first of all, monocyclic or single cycle pathogens, these pathogens complete one disease cycle in 1 year or per season. So for example, smut of sorghum or kambu, root rots of pulses and wilt diseases. So there is no secondary inoculum and secondary infection. So it complete their life cycle with their primary inoculum itself. So these pathogens are mostly responsible for slow epidemics. So it produce disease in slow epidemic manner. So graphically these pathogens represents a saturation curve. Saturation curve means decreased curve. It will produce decreased curve. So next one is polycyclic or multicycle pathogens. So it will complete more than one disease cycle per growing season. For example, rust, powdery mildew, leaf spot, leaf blight, anthracnose, etc. So the amount of inoculum increased many-fold in particular crop season. In this polycyclic pathogens, disease initiated by primary inoculum and it will produce numerous secondary inocula. So inocula is a plural of inoculum are produced at each infection site. So mostly cause rapid epidemics in field. So it multiply the disease in rapid manner or in continuous manner. Graphically these pathogens represents a sigmoid curve, so increased curve. It will produce increased curve. So last one is polytic pathogens take more than 1 year to complete a single disease. So for example, Dutchium, Citrus tristissa, pure decline. So Citrus tristissa is a viral disease. It take more than 1 year to complete their life cycle, and it continuously multiply their progenies in particular host, may cause severe epidemics over several years. So various stages in pathogenesis. So first of all, inoculation. Inoculation means the pathogen come into contact with a particular plant, means that is called as inoculation. Next one is prepenetration. Prepenetration means in this stage a pathogen is ready to penetrate into the pathogen. And next one is actual penetration stage. Next one is infection stage. In this stage the pathogen take nutrients from plant and multiply itself. Next, dissemination of the pathogen. In this step pathogens disseminate from infected field to non-infected field, or infected plant to non-infected plant. Last one is overwintering or oversummering. After end of particular crop season, the pathogens overwinter or oversummering on another source, that is soil, plant debris, or weed crops. So in penetration there are three types: direct penetration, penetration through wounds, penetration through natural openings of plants, for example stomata, hydatodes, lenticels, etc. So the first two steps, that is inoculation and prepenetration steps, are basement steps. In these steps pathogen only put basement on plant surface or plant organs to enter into the plant. So before see about inoculation, we should know about these terminologies in plant pathology. So first of all, inoculum, any part of the pathogen that can cause infection or infective propagule coming in contact with the host. So inoculum in the sense, any part of the pathogen which have the ability to infect particular host. So the propagule means one unit of inoculum of any pathogen. So a small unit or one unit of inoculum of particular pathogen is called as propagule. Next one is primary inoculum. So a inoculum that survives dormant in the winter or summer or in unfavorable conditions and initiates the infection first time in the season of crop. So the role of primary inoculum is initiate the disease in a particular area which don't have any diseases earlier. So next one is secondary inoculum. An inoculum produced from primary infections is called as secondary inoculum. So primary infections produced by primary inoculum. So primary infections produce the secondary inoculum of a particular pathogen. It helps in disease epidemic. So secondary inoculum multiply the or spread the disease from one plant to another plant or one field to another field. So inoculum potential, the amount of inoculum responsible for successful infection. One successful infection is called as inoculum potential. So the minimum amount of inoculum or part of pathogen to produce a single disease or single infection is called as inoculum potential. So first step is inoculation. In this step the pathogen coming in contact with the host, that is susceptible host. So inoculation occurs through these several things, that is first of all plant debris. So the inoculum may be overwintered in previous season crop debris. So crop debris and also soil of particular field, and also through planting material like scion materials, grafting, cuttings. So next through seeds. So seeds are one of the important transmitting material of disease. There are two types of seed-borne pathogens. First of all, externally seed-borne pathogen. So pathogens only present on seed coat. It does not enter into the seeds. So second one is internally seed-borne disease. It mainly infects the embryo or it infects the internal tissues of seeds. Next one is the inoculation may be by passive things, that is air, rain water or rain splashes. Next through vectors. So vectors play an important role in inoculation. The virus, bacteria and viroids are mainly transmitted through these insects. So you can see the some of the inoculums. In the case of fungi, transmit the disease through their inoculum, that is mycelium. So first picture represents the mycelium. Mycelium is nothing but group of fungal bodies. Group of fungal body is called as mycelium. The group of fungal bodies act as inoculum for particular disease. So next one is next picture represents the spores. So inoculum may be the sexual or asexual spores of fungi. Sexual spores means the spores produced by sexual reproduction, and asexual means the spores produced by asexual manner. In contrary, for bacteria, protozoa, spiroplasma, phytoplasma and fastidious vascular bacteria, the inoculum is always a whole individual of particular pathogen. For example, protozoa means the whole individual act as inoculum for particular disease. So this is your protozoa. Next picture is your bacteria. So different types of bacteria, plant bacteria. So next one is topmost picture is represents the phytoplasma, candidate is phytoplasma. Next one is spiroplasma, helical structured bacteria and also wall-less. Next one is different types of viruses. Viruses also act as inoculum. Whole individual of single virus is act as inoculum. And the next one is naked virus, or also called as viroid. So these are all the inocula for plant diseases. So next one is second event or second step, prepenetration. In this stage, pathogen is present on the host surface but has not entered into it. So it does not enter into plant, but it put the basement on particular plant part. In this stage, you should know about infection court. Infection court means the site of host parasite relationship, initial host parasite relationship in first infection. So this is your infection court, the host parasite relationship in first infection. So in this stage, pathogen produce the myceliaceous substance to adhere the plants. So it will produce the adhesin chemicals to adhere to the plant or attached to the plant. So next one is in stage 1, it will produce the aprosorium. So aprosorium is a specialized structure produced by fungi to attached with or adhere with plants. This is also called as anchoring organ. So it will anchor the plant. So the swollen tip of germ tube that facilitates attachment and penetration of the host by fungus. So mainly for attachment to the plant. So you can see this picture. In first stage, conidium is a spore of fungi. The pathogen coming in contact with the host surface, and it will produce germ tube. Germ tube is nothing but the first hyphae or first body of fungi produced from spores. Spore germination is like seed germination. It will produce germ tube. After some time, the tip of germ tube bulged and produced aprosorium. So this aprosorium gives the pressure against plant surface or cuticle and cell wall of particular plant to puncture the particular plant pot. So third step is important step, that is actual penetration. So penetration means actual entry of pathogen into the host plant. So based on their capacity of enter themselves into the host cells, pathogens are classified into active invaders and passive invaders. So the pathogens invade themselves means that is called as active invaders are active pathogens. In contrary, pathogens need some help from external agencies means that is called as passive invaders. So active invaders, pathogens which make an aggressive effort to gain entry into intact host cells and do not require help of any external agencies like insects, etc. So passive invader means pathogen which do not have any capacity to enter the host cell or puncture the host cell. So it mainly enters through some vectors or external agencies. It requires some external agencies like insects, wounds of plants or natural openings of plants. So for example, virus, viroid, protozoa and vascular bacteria are the passive invaders. So direct penetration is the example of active invader. So pathogen require adhesion to the plant surface allowed by the application of pressure and enzymatic degradation. So in direct penetration, pathogen will produce enzymes, degradation enzymes like also called as lytic enzymes like cellulase, pectinase to puncture or to dissolve the cellulose and pectinose. So pectin contents. So this pressure is aggressive pressure is given by that swollen region, that is aprosorium, cuticle and cell wall, in order to overcome the physical barriers of plant surface. So next one is penetration peg. So penetration peg or infection peg is a small finger-like growth from underside of aprosorial cell. So aprosorial cell will produce the small finger-like peg to penetrate the cuticle or cell wall. That is called as penetration peg. Then Hastorium is a specialized structure produced by pathogens or some fungi to suck the nutrients or drink the nutrients from plants. So Hastorium is simply called as drinking organ of fungi or sucking organ of fungi, mainly produced by obligate parasites, that is erysipelas and puccineals. Fungi belongs to erysipelas and puccineals. So the word ends with ales means that is denote the order of fungi. So erysipelas and puccineals are two order of fungi. Fungi belongs to this order, produce the Hastorium. So next, second stage of penetration, after 8 hours of inoculation, the aprosorium produce the penetration peg and enter into the plant. After 10 hours, elongation of penetration peg will occur. Penetration peg will elongate into the plant. Then stage 3, at this stage, penetration peg produce the membrane of Hastoria. So initiation of Hastoria, it will produce Hastorial initiation steps. Next, after 14 to 15 hours, young Hastorium will be produced in host cell. Next, stage 4, after 16 hours, the matured Hastorium produced by our fungi, and it will it ready to take nutrients from plants. So you can see the all 4 stages in this picture. First, the plant come in contact with the plant surface, that is inoculation. Then it will produce germ tube and Aprosorium. In second stage, it will produce penetration pip. Third stage, it will produce young Hastorium, that is drinking organ. In fourth stage, Hastorium will mature and ready to take nutrients. So this picture represents the spore germination and penetration in clear way. You can have a clear idea about spore germination and penetration in this picture. So first of all, spore germinate and produce germ tube and produce Aprosorium, that is bulged appearance or swollen organ to attach with the plant, and it will give the pressure to pressure against the plant parts, that is cuticle and epidermis. After some time, Aprosorium produce the penetration pig. It will puncture the cuticle region. This is your cuticle region. Puncture the cuticle region and produce penetration hyphae. So hyphae is nothing but a unit of fungi. So single unit of fungi is called as hyphae. Penetration hyphae invade the host cell. Invasion means spread into particular tissues or particular plant tissues. So the hyphae invade into particular plant tissue and produce Hastorium. So Hastorium is a drinking organ. It enters into epidermal cells and take nutrients from them. So this is your epidermal cells. So this is also one of the picture represents the spore germination and penetration. After penetration, it will multiply the spores. So these are all the examples of direct penetration. First of all, Plasmodia fora brassicae. This is the protozoan pathogen. It will produce clubroot disease. It mainly infects the root tissues. Infected root tissues bulged due to hypertrophy and hyperplasia. Hypertrophy means multiplication of enlargement of cells. Hyperplasia means multiplication of cells through increased number of cell division. So next one is Pythium species. This is damping of pathogen. It mainly infects the seed coat and the emerging seedlings. The nature of pathogen is it collapses the collar region of plant at their seedling stage. So next one is Istulago tritici. This is mud fungi, loose mud fungi. It mainly infects the stigma and the ovary walls. So it converts the whole seed into black color spores. So we can't take the raw spore, or we can't use that infected spores to edible infected seeds to edible purpose or seed purpose. So next one is claviceps. Claviceps is a ergot fungi. Ergot is also called as sugary disease. It mainly infects the stigma and it will produce sugar-like substance or honey-dew-like substance on the infected portion, mainly in flowers of monocotyledons like sorghum, kambu, etc. So next one is Ericifae species, one of the important genus in fungi. So it is a powder mildew fungi. It produces powdery growth, white color powdery growth on upper surface of the leaves. So it mainly infects the epidermis layer and take nutrients from them. So Ericifae mainly produce Hastorium. Next, Phytophthora infestans. This is a pathogen, a historical pathogen. It caused the Irish famine in 1845. So in Ireland. So the disease is layer plate of potato. It mainly infects the cuticle and cell wall and produce necrotic regions on potato and potato leaves. So next one is Magnoporthygriaceae. This is also a historical pathogen. It causes the blast disease, rice blast disease. It puncturing the cuticle and produce the eye-shaped or spindle-shaped spots on infected leaves. Next one is Penicillium botrydiplodia. These are all the fruit rot fungi. It mainly infects the stalk end. So methods of penetration and invasion by fungi. So all three penetration methods of fungi represented by this picture. First of all, direct penetration. There are several direct penetration methods. First of all, direct penetration with the hosturia. So it will produce hosturia and take nutrients from them. Next, direct subcuticular only. So directly infect and it will not enter into the epidermis layer. Only survives in subcuticular region and take nutrients from them. Next one is direct with aprosorium, penetration peg and intracellular mycelium. It will produce intracellular mycelium. So what is meant by intercellular and intracellular? Intercellular means the pathogen present in between two cells, that is called as intercellular. So in contrary, intracellular means pathogen present inside the cell. So whole pathogen present inside the cell means that is intracellular. So in this case, pathogen present inside the cell, that is intracellular. So next one is direct and intercellular mycelium. In this case, pathogen does not enter into the cells, but present in between the cells. You can see this picture. So next, direct intercellular mycelium with hasturia. So intercellular mycelium, and also it will produce drinking organs. You can see that. Next method is penetration through natural openings. So natural openings in the sense, the important natural openings of plants are stomata, lenticels, hydathodes. So stomata is a natural opening of plant, particularly present in lower side of leaves. It will open at morning time and more or less closed at evening time. So through this opening, our pathogen easily enter into the plant. So next one is through lenticels. Lenticels are raised pores, raised hole appear in our plant bark or woody bark. It will also for gas exchange. So next one is hydathode. Hydathode is also a pore present at leaf margin to excite the extra water at the time of high humidity, like our sweating. So next one is through penetration through natural wounds. So through natural wounds, and also enters through cracks between main and lateral roots, and the infected fungus kills and macerate the infected tissues through enters through wounds. So there is no pathogen does not produce any aggressive pressure against natural openings. So it will nicely enter into the natural openings already present in plants. You can see this picture. Otherwise, all the processes are same as direct penetration. It will produce hasturim and take nutrients from plants. So example for natural pathogens which enter into the natural openings are stomata. So rust, downy mildew, powdery mildew pathogens are mainly enters through stomatal region. Usually bacteria, all the bacteria, hydathodes and wounds. Then lenticels, bacterial powdery scab of potato, that is Spongospora subterrana, and root rot of apple, Armillaria mele, apple stem canker, Nectria species are mainly enters through lenticels. And fireblight of apple, so first described or first reported plant bacterial disease caused by Erwinia amylovora, enters through nectaries of flowers and plants. So this picture represents the methods of penetration and invasion of bacteria. So bacteria mainly enters through natural wounds and natural openings. Why? Because there is no specialized organs in bacteria. So through stomata, through natural wound, also through hydathode, and also through nectars present in flowers. So fourth step is infection, one of the important step in disease cycle. In this step, establishment of pathogen within susceptible host. So after entering to the host, that is penetration, after entering to the host, pathogen will take nutrients from plant and establish or multiply their progenies in particular host. Susceptible cells, tissues of the host, and starts to procure nutrient from them. So infection period, so time elapsing between spore germination and established infection. So the time period between spore germination and established infection is called as infection period. So at the same time, invasion means pathogens spread into all the tissues of plant organs, or in particular pathogens invade the all tissues, that is called as invasion. The pathogen and fungi and bacteria mainly through inter or intracellularly, virus, viroid, fastidious vascular bacteria mainly through intravascularly. So these are all the pathogens present inside the vascular tissues, that is xylem and phloem tissues. So there are two types of infection, local infection and systemic infection. Local infection means the affecting pathogen produce limited symptoms, and also the affecting limited part of plant, or it only infect the contact region. For example, fungi, except smut and few down mildew fungi. For example, Cambu down mildew is a systemic disease, so we can't include the down mildew fungi in local infection. And bacteria, some bacteria, and phanerogamic parasites like Cuscuta, Loranthus, Orabanchi, Astriga are the examples for local infection. It infect the particular area of plant. It can't move within the plant. So in contrary, systemic infection means single infection point to whole plant or organ. So particular pathogen spread from single infection point to whole plant. For example, HIV. So they remote in their action. So it can spread from a single point to whole plant, whole part of the plants or whole organ of the plant. For example, virus, viroid, fastidious vascular bacteria, candidatus phytoplasma, and protozoa. So next one is colonization. Like human colony, pathogens also invade the plants and take nutrients from them and starts to multiply. It will colonize with small group of or number of pathogens. So that is called as colonization. Next, fructification period. Fructification means the time gap between or time period between infection and first reproduction. So after infection, when the pathogen starts to reproduce means that is fructification period. Infection and first reproduction. Time period between infection and first reproduction is fructification. So the successful infection results in appearance of symptoms. So particular disease, each and every diseases will produce symptoms, particular symptom to exhibit the host reactions. So incubation period, time between inoculation and system expression is called as incubation period of particular pathogen. So this is about your infection. So main role of infection is take nutrients from plant and multiply into them. So fifth step is dissemination of the pathogen. So dissemination is nothing but transport of inoculum from the source of production to an area where a susceptible host is growing. So dissemination means transport of inoculum from infected source to non-infected source, or infected field to non-infected field, or infected area to non-infected area. So there are two types of dissemination in pathogen: active dissemination, passive dissemination. So active dissemination mainly through plants, the transportation of inoculum through plants, plant pots, plant debris, soil, and seeds. These are all the active disseminations. So passive dissemination means there is an involvement of external agents like insects, air or wind, irrigation water, and rain splashes. These are all the external agents help to pathogen to disseminate the pathogens. So virus, viroid, fastidious vascular bacteria, phytoplasma, and spiroplasma. These are all the pathogens multiply themselves in particular living tissue and mainly transmit through external agents and plasmodesmata. So sixth event is overwintering, oversummering, and survival of pathogen. So in this stage, inoculum survives in debris, soil, or seed, etc. Because of their absence of host. So after end of crop season, the pathogens overwinters on debris, soil, or seed, etc. So obligate parasites survives in living tissues. So in contrary, obligate parasites only present in living organism because it only take nutrients from living organism. So it survives in another host like weed host. So after overwintering, the pathogens at the time of overwintering, pathogens have hard structures to survive the unfavorable, to tolerate the unfavorable conditions. So this is about your pathogenesis. So this is a cyclic process. Pathogenesis is a cyclic process. So it will occur in cyclic manner. So the overwintered pathogens will germinate at the time of new season or new susceptible host. So this is about your pathogenesis. Thank you.